The precise spatial-temporal regulation of genes is controlled by diverse cis-regulatory elements that interact between them. Our protocol makes it possible to quantitatively interrogate chromatin contacts of selected loci, such as promoters. This technique allows us to identify and quantify the interaction frequencies between various regulatory elements simultaneously.
To generate embryoid bodies, begin by culturing mouse embryonic stem cells to 60% confluency. Then, remove the culture medium and wash twice with 2 ml of sterilized PBS. Remove the PBS completely and add 2 ml of cell detachment medium. Incubate the culture dish at 37 degrees Celsius. After incubation for 5 minutes, add 8 ml of EB differentiation medium to the dish. To dissociate the MESC colonies to obtain a single-cell suspension, pipette the medium up and down 15 to 20 times and transfer the suspension to a 10 ml tube. Centrifuge the cells at 300 times G at room temperature for 5 minutes. Then carefully remove the supernatant. After counting the cells, resuspend the cell pellet with EB differentiation medium. Invert the lid of a 15 centimeter culture dish. Using a 200 microliter multi-channel pipette, deposit 20 microliter drops of resuspended cells on the lid. Invert the lid carefully and place it onto the bottom portion of the culture dish.